All right. Hello, my name is Hadi Ammar. I go to Polytechnic School, and I'm interested in the study of genomics, especially microbiomes, and I'm part of the Goentoro Lab Group. So, before I share my experiment, I'm going to give some insight into why I did my experiment. So, about 2 years ago, the Goentoro Laboratory published a paper in which they noticed a conserved stimulus of post-amputation regeneration in flies after treatment of leucine, which is an amino acid, and insulin, which is a growth hormone. And so as you can see, the control here, there's no regeneration, but in 3 special cases, in the image right here, you can see regeneration after the amputation site. And also recently, this is unpublished, but one of my lab mentors, Zevin, noticed that after feeding flies a bacteria known as Lactobacillus brevis, after amputation, the flies would actually have a muscle survival. So as you can see in the control, after amputation, the muscle dies. So there's no light at all. It's completely dark, but in the treated samples, there's still some muscle tissue surviving near the site of amputation. So after L. brevis is ingested, we assume that the bacteria interacts with the microbiome within the fly gut. So as you guys may know, there are bacteria in all of our guts. So we want to know why the interaction between Lactobacillus L. brevis and the other bacteria cause this muscle survival. So my goal in my experiment was to characterize the fly microbiome. And I want to know what's in the microbiome to better get an understanding of how these interactions happen. So here's how I did that. The first step is kind of sad, but we blended up some flies. It's called, the step's called homogenization. And then we took the supernatant, which is just full of the bacteria in the gut, and we plated it onto 2 different types of plates. So one that's selected for lactobacillus bacteria, and the other one selected for acetobacter. So after that, we picked phenotypically unique colonies that looked unique, and we plated them onto other plates. And so we did this for 3 rounds until we had about 80 samples. So after we had those samples, we put them into glycerol stocks to maintain them for a long period of time. Then we transferred them into liquid cultures to better access their DNA. Because the goal here is to sequence the bacteria to know what they are. And yeah, so next we did DNA extraction using a homemade kit that we found in a textbook in our lab. So we just set all that up. Anyways, we got the DNA from that extraction experiment. And then next we ran the DNA through PCR DNA amplification to get a higher concentration of DNA so they could be sequenced more effectively. And then next we went through gel validation and extraction. So the way you do this is basically you put all the DNA through gel electrophoresis, and then you basically excise the DNA that corresponds to the number of base pairs that you would see typically in bacteria within the gut. And next you can basically purify the DNA so that it's ready to be sequenced. So this is our actual gel. And so this is after PCR, like I said. So these 2 circled gels right here are corresponding to 1,660 base pairs. So those are bacterial DNA, but the 3 on the bottom are primer dimers. So this results in an error or malfunction in the PCR stage. So that's basically a contamination that may have resulted in a failure of sequencing later on. So the way we can fix that is by purification. So when you basically separate the different base pairs by even a larger, you separate the gels by even a larger distance by running it through the gel electrophoresis, and that way we can better excise or take out the DNA that only corresponds to the bacteria. So we sent in our bacteria for sequencing, and it came back 2 days ago, and unfortunately it was rejected. But we have about 60 more samples to send in, so we'll be doing that soon. Yeah, thank you all for listening, and I want to say thank you to my lab mentors for answering a million questions for me. So yeah, thank you so much.
Have any questions? So the application for this, in terms of
so
generating
basically exactly. So like regeneration of limbs in humans, getting a better understanding of how that happens in flies to start out, so we can eventually apply it to humans as well in the future.
Probably a long timeline.
Yes.
Because the lab has been working on this for quite a while.
Exactly. Yeah. All right. Thank you.
So you were a group of 1.
Yes, I was
you
group of
didn't
1.
have the camaraderie of other students.
No.
You had more mentors per 1 student than most groups.
Exactly. Yeah.
I'll ask you a question. So
Yes.
how does it know to regenerate it? There's a DNA that has the information and what is being regenerated and it's like a new animal being born and making a new limb, so that blueprint is still
Yeah, that's
there.
like a great question. So like initially, like the lab thought had something to do with like a metabolic pathway. So like insulin and as well as like leucine, like they could both like metabolic signals. So and like another hypothesis that we had was that had something to do with like the microbiome. So it might come from both areas, but we're not specifically sure which one it is. But we know that both of them have effects of regeneration. So that's still like an ongoing answer that we're trying to figure out. Yes.
Does the injury itself have something to do with reprogramming?
What was that? Sorry.
Like the injury itself, whenever you're doing an amputation, does that actual force have something to do with reprogramming of local cells and that it could form like a blastema or something?
I'm not exactly sure how that works, but I would assume so, especially because like in like the picture demonstrated from my mentor Zevin, they're like because like usually like the muscle tissue will die like surrounding like the side of amputation, but in this case there's survival. So I assume that there would be a signal that would basically like signal like an apoptosis of like the muscles near like the amputation versus like that signal not being sent when there is L brevis in the system, but I'm not exactly sure how that works.
I think people have at least one thing that can regenerate, and that's the liver. If you hack away 90% of the liver, it'll grow back.
That's cool.
Any questions? Thanks again.
Thank you.